Hybridoma is aimed at industrial production of monoclonal antibodies. The first process in making a hybridoma is to generate antibody producing B cells. This is done by injecting a mouse with an antigen of interest, thereby provoking an immune response. Multiple injections of the antigen of interest are carried out over a period of weeks until an appropriate antibody titer is achieved. To determine if the mouse is producing antibodies of interest, test bleeds are performed and examined for the presence of antibodies. Some of the B cells will produce the antibodies that bind to the epitopes on the antigen of interest, shown in blue and red. Others will produce non-specific antibodies that do not recognize the injected antigens. If the host is producing the desired antibody, the splenocytes are harvested. This is done by the removal of the spleen and dissociation in culture medium to release the resident B cells. The culture medium also includes cells from a special mouse myeloma cell line. These tumor cells can divide indefinitely but do not produce antibodies. When polyethylene glycol (PEG) is added to the mixture, some of the two types of cell fuse to form hybridomas. The next process is to separate the fused hybridoma cells from the unfused B cells and myeloma cells. Unfused B cells will die because they lack the ability to survive in culture, leaving only the hybridomas and unfused myeloma cells. The myeloma cells lack HGPRT gene, causing them to die when placed in a hypoxanthine aminopterin thymidine containing medium or HAT. In this medium, only the fused cells survive because the B cells contribute to the functional HGPRT gene. Basically, the hybridomas survive in culture because of its myeloma properties and is protected from HAT selection by its B cells properties. Surviving hybridomas are separated and individually cultured, one cell per well. These cells are described as clonal cultures because all the cells from each well are derived from a single cell and are therefore identical or clonal. After culturing for a few weeks, when the growing cells can be seen, the culture fluid can be screened for the presence of desirable antibodies. Screening is the most labor-intensive step in the production of monoclonal antibodies, since fusion can result in thousands of the individual hybridoma colonies. Note that different antibodies may react to different epitopes on the same antigen. The formation of a colored product indicates a positive hybridoma. So finally, clones that produce the desired antibody are grown in mass culture and are frozen at -20 degrees Celsius or lower for storage.